So with T cells, since they can't recognize pathogens in general, they have to have them associated with our body cells. We have special proteins on our body cells that can interact with antigens. They're called major histocompatibility complexes. We have two types. Type 1 is found on every one of our body cells. They're basically what give each of our cells a fingerprint of its own. And none of us have the same type of MHC1 proteins on our cell surface. The only types of cells that don't have these are cells that don't have a nucleus and those organelles. So like red blood cells aren't going to have MHC class 1 proteins on them. The rest of our cells do. And so when our cells get infected with a pathogen, they can break down that pathogen and they can incorporate their self-protein and the pathogen together and present it on the surface. And then our immune system cells can respond, or our T lymphocytes can respond. We have two types. I told you type 1 is MHC class 1. And then we have these things called antigen-presenting cells, which have another MHC protein called MHC class 2. So if you're an antigen-presenting cell, you have both class 1 and class 2. MHC class 2 are found only in phagocytic cells. And we only have three types of cells that we typically talk about. We talk about dendritic cells, macrophages, and B lymphocytes that are antigen-presenting cells. And antigen-presenting cells will phagocytize material, and then they will present the pathogen on the surface of their cell with the MHC class 2 molecule. So different T lymphocytes can respond to different antigen-presenting cells. Different T lymphocytes can respond to different types of MHC complexes. MHC class 1 is going to be interacting with cytotoxic or killer T cells. MHC class 2 interacts with helper T cells. And I just talked about this, so this is just another page saying exactly what I told you. And I'm going to go through and, I'm going to move from this slide because this has information that you can read, but I'm going to explain it on the next slide, okay? So here we have our cell itself, okay? And inside of the cell, we have all of our different organelles. And so here we have self molecules that move into the rough ER. And in the rough ER, we produce proteins. So these self molecules and these proteins that we produce come together, they bind together, and then they're put into a vesicle, transported to the Golgi, and the Golgi then will move them to the plasma membrane. So they become part of that plasma membrane. And so here's what it looks like. So this is just a basic MHC molecule with a self-antigen associated with it. This is on all of our body cells, so these would be MHC class 1. These are the ones that our cytotoxic T lymphocytes are going to react to. So if we get infected with a viral infection or something, then some of the viral proteins can be degraded and can be inserted into the self-antigen. So here we have that viral protein coming in, or the viral material coming in. We break it up and when we break it up, we then insert the viral material with our self proteins, and that's going to be with our self proteins, and that's going to be attaching to the MHC class 1 molecule. And so that's going to be presented on the surface of the cell, and now we have self and non-self together. And when our T lymphocytes attach and they see that it's self, non-self, they're going to stay bound and they're going to get active or become active. MHC class 2 are a little bit different. They're only on antigen-presenting cells. And they're going to be presented if the pathogen, or if a pathogen is taken in and broken down by a phagocytic cell. So we have some phagocytic cell that gets taken in, and here just bacteria, that's going to be broken down. We have these MHC class 2 molecules here that are produced, moved to the Golgi, and in the Golgi, they're put in a vesicle. The vesicle here has that class 2 molecule, and then we have this phagolysosome here that is being broken down. And what we produce is we produce particles here that come in contact with the MHC class 2 molecule, because here you have that lysosome releases chemicals, breaks down this molecule, and so you end up having these non-self proteins associated with this MHC class 2 molecule that is then put on the surface of the cell. And helper T cells are going to interact with that one. So we have cytotoxic T lymphocytes interact with MHC class 1. Helper T lymphocytes interact with MHC class 2 molecules. So, like I said, MHC molecules are unique to each individual. Unless you're an identical twin, then you would have the same MHC molecules for identical twins. But otherwise, typically we have unique MHC molecules. And this causes problems because our immune system knows the difference between our MHC molecules and non-MHC molecules. And because of this, organ transplants are more difficult. So they have to try and match people up very effectively, because you first have to take the patient's MHC molecules, and then the donor's MHC molecules, if they're similar enough, they may have our immune system, be, what's the word I want? Our immune system may not recognize the difference, but in general it will. And so what they do is they get very similar donors, and once they have the similar donor, they're then going to start releasing or giving immune suppressant drugs, so our immune system is suppressed so it doesn't attack. Even if it's very similar, you don't want it to attack the transplanted tissue. And the hope is that your body will incorporate that tissue into itself, and eventually you will be producing new cells that have yourself on it. So now let's talk about what goes on in the life of a lymphocyte. So lymphocytes can live for a good amount of time. Once they become activated, then they're not going to live much longer, because they're going to attack and they're going to fight off the infection, and then they're just going to die. But up until they get activated, they kind of have a nice little life. They are born in bone marrow. All cells, all blood is produced in bone marrow. T lymphocytes move to the thymus, where they are taught the difference between self and non-self. B lymphocytes stay in the bone marrow. where they're taught self from non-self, and then they become immunocompetent and they move out into our lymphatic system, go to lymphatic tissues, they reside there until they come across the first exposure of an antigen. Then they get activated, and then usually once they're activated, a few cells are going to remain behind. Those are your memory cells. And you have your active cells go out and they fight off the infection. And that would be that effector response where they're fighting off the infection. And B lymphocytes produce tons and tons of antibodies. T lymphocytes fight off the infection directly with the cells that are infected. And so here we have bone marrow birth center where all our blood cells are produced. And then some of those blood cells move to the thymus if they are T lymphocytes. If they are B lymphocytes, they stay in the bone marrow. They go through a maturation process, depending, again, T lymphocytes in the thymus, B lymphocytes in the bone marrow. And once they become immunocompetent, they leave the thymus or the bone marrow and they move to their lymphatic tissue that they're going to reside in. And so here we have T lymphocytes being produced and B lymphocytes being produced. The pre-T cells move to the thymus. The B cells, pre-B cells, if you want to call them that, stay in the bone marrow. Once they go through their positive and negative selections, then naive immunocompetent cells are going to leave and move to their lymphatic tissue that they're going to reside in. So there's two different tests that these cells are going to go through. Positive selection is a test where we have these T lymphocytes that have to be able to bind to major histocompatibility complexes. So our T lymphocytes, pre-T lymphocytes, have both types of CD receptors. So they have CD4, CD8. They have other CDs, but we only talk about those two. And in the thymus, these cells have to be able to attach to either MHC class 2 or MHC class 1 molecules. If they can't bind, then they can't initiate an immune response and they're going to die. If they can bind, then they survive. From there, we have a negative selective test. Any T lymphocyte that binds and stays bound is going to be killed off. Any T lymphocyte that binds and then unbinds immediately because it doesn't recognize danger is going to survive. So positive selection, you have to be able to bind. Negative selection, you better not stay bound. If they pass both of these tests, then the T lymphocytes that have passed the test will survive and move on. Any other T lymphocytes, if they did not pass, are going to be killed off. Once they've passed the tests, both tests, then they're going to lose whatever receptors that they don't need. So if you're a cytotoxic T cell, you're going to lose your CD4s. If you're a helper T cell, you lose those CD8s. And now you're considered a mature, naive, immunocompetent T lymphocyte. B lymphocytes have a similar process. And I'll talk about that in a second, sorry.